essentially throughout mammalian evolution, almost as if these two amino acid residues are the only residues that are actually permitted at this particular position, but changing from one to the other and perhaps back again can completely change the repertoire of your escape from these mimic antagonists that are encoded. So we might actually have reached some remarkably general conclusions about host proteins and how they might overcome mimicry by pathogenic viruses and bacteria. So the first important rule is you definitely need evolution on multiple surfaces. If all you had was the alpha G evolution, it would still be a very easy task for K3L, whose only job is to look like EIF2 alpha, to basically block this interaction. Instead, what PKR evolution seems to reveal is that the first step in their evolution is paradoxically a negative affinity step, where they're moving away from binding both EIF2 alpha and consequently K3L, and then restoring its binding to EIF2 alpha specifically by virtue of changes at a different surface. And that means that not only are multiple surfaces rapidly evolving, there's extraordinary flexibility that's been actively selected on PKR just by virtue of the challenge that it faces because of mimicry. Just to show you an example of this negative and positive affinity, here's again that example of orangutan PKR, which is able to defeat K3L. But you notice that the orangutan G helix, which is the most important determinant of EIF2 alpha binding, is actually not doing such a good job when it's in a chimera in a gibbon backbone, which actually means that even though orangutan PKR can actually bind EIF2 alpha really well, its G helix is somewhat compromised in terms of its binding to EIF2 alpha. And indeed, epistasis of a different part of the orangutan backbone has actually successfully restored the complete binding and recognition of EIF2 alpha. So we have these states where you're moving away from affinity and binding again, and you're basically cycling between these states as a way to overcome this mimicry interaction. What that means in terms of overcoming mimicry for PKR versus the other three EIF2 alpha kinases encoded in vertebrate genomes is that unlike these guys, which are all evolving under purifying selection, and they have basically optimized for their substrate recognition of an unchanging EIF2 alpha, PKR cannot afford to get this optimized for substrate recognition, because as it gets better and better for substrate recognition, it becomes more and more of a sitting duck for the mimic. So instead, it's actually actively engaged in this selection exactly in a suboptimal substrate recognition in order to gain maximal discrimination against these mimics, which means that in terms of its core housekeeping function, which is EIF2 alpha recognition, the mimicry has forced PKR to become slightly suboptimal. Why is this sort of an important and profound insight? We think it's important because if you think about every single cellular process in the cell, cytoskeleton, membrane trafficking, cell cycle, and apoptosis, for instance, all of these processes are actively shaped currently in human populations by active viruses and bacteria, which means that the alleles that we see in the human population for these absolutely essential housekeeping processes need not be the alleles that are best at their housekeeping function. What might have actually survived in the course of evolution are the best compromise alleles that had at least some housekeeping function, but were able to actually overcome the challenge imposed by these pathogenic mimics. So in a sort of a ironic way, these evolutionary winners may in fact be losers in terms of their housekeeping function. So I'm going to actually switch to the viral side of this conflict for a second to sort of try to understand why K3L cannot do a better job of being a mimic. I'm not actually going to answer that question completely here because we don't fully understand why K3L is not taking advantage of the situation that seems to be set up for it. But one important clue that's already emerged is that K3L also is acting under dual constraints because it needs to mimic the PKR interaction interface, but cannot actually touch the other EIF2 interaction interfaces, and that's the challenge that it's basically facing. But we took advantage of this system to ask a slightly different question, which is, how do pox viruses evolve so rapidly at all? Unlike RNA viruses that you've heard a lot about, like polio and influenza, which evolve so rapidly that they can explore mutational space very quickly, the rate of poxviral mutation is actually almost on the order of eukaryotic genome evolution, so very slow to evolve, and yet they can be some of the most successful pathogens among mammals. So we took advantage of this PKR and K3L system to set up a situation where we could look for poxviral adaptation and maybe uncover methods of their adaptation. So in this pathway of double-stranded RNA recognition and squelching by E3L and the K3L antagonist of PKR, if we now delete this E3L gene from vaccinia, putting all the selective pressure on K3L, we've already established that the vaccinia K3L is really poor against human PKR, and as a result, you basically drop 3 logs of fitness. Basically, that's a huge deleterious advantage when you passage in human cells. So what Nels did was he took these viruses, which were somewhat debilitated, and simply passaged them in HeLa cells at a low multiplicity of infection in replicate populations. And because we are evolutionary biologists, we basically kept a fossil record of this virus, just so that if adaptation occurred, we could go back to our freezer and understand what might have happened. So when we passaged these, we were basically ready to do this experiment for about a year. But 10 passages into the experiment, Nels came to my office and showed me this graph. So this is, again, the viral fitness of the wild-type vaccinia virus that has E3L. This is the parental virus of our replicate populations, which is delta E3L. You can see a huge drop. And in just 6 passages, you can see that these viruses have basically regained most of the fitness that they have lost. Now, this is a glass half empty, glass half full situation. If you're an evolutionary biologist, you're delighted, because clearly adaptation occurred, and you can now study it. If you're an epidemiologist, however, you're a little concerned, because this is one of the most potent defenses that we have against these viruses, and it literally took 6 passages, which is less than 6 weeks, for the virus to basically evolve a potent solution to this. Here's another way we can actually look for viral fitness, which is replacing the E3L gene with beta-galactosidase. We can do a simple mug assay to look for fitness, and that also shows that the replicate populations, all of them have basically increased in fitness. So we turn to the dark power of next-gen sequencing to tell us magically what happened, and so maybe this would solve our problems. And I have to tell you, the initial returns were not very impressive. It was sort of downright depressing, because after 10 passages in all of the 3 replicate populations in a 200 kb vaccinia genome, these are all the 3 mutations that we basically found. This one turned out to be an artifact later. So what that means is that the HeLa cells that we were passaging these viruses in have a higher mutation rate per base pair per genome than the vaccinia viruses that we were going. So these are really slow viruses. I'm going to come back to this change a little later, but clearly this change was not frequent enough or in all 3 replicates to actually explain the adaptation. So we then turned to the copy number variation that was evident in the terms of numbers of reads that we could see in these vaccinia. And when we line these up to the parental genotype, we see 2 remarkable things. The first is that all of these parental and replicate viruses drop to a genomic coverage of 0 exactly at the E3L locus. Now, this is exactly what you should expect, because all of these have been deleted for the E3L, so they don't have anything to line up to. Also showed us that we did not have a contamination issue. But the more interesting thing happened with this blip right here, which basically represents a copy number expansion of all 3 replicates independently through completely different breakpoints compared to the parental virus, which basically has a genomic coverage of 1. Which means that in every instance that we have looked at, we've done this experiment 6 more times, the initial step that vaccinia takes when faced with a PKR it cannot overcome is copy number expand the K3L locus to basically try to overcome that. So we thought that we basically have now viruses which had a triplication or quadruplication that have now taken over in a classic Darwinian way. And again, we were slightly wrong, because when we went to a southern blot looking at this sort of viral population, remember we have kept now a fossil record, and you can see starting from the parental delta E3L virus, as we go up in size, in passage number, you start seeing this sort of accordion-like expansion of K3L copy number appear. And this copy number is still on the way up. So which means that at passage 10, we have an average copy number of 4, but there are viruses all the way from a single copy all the way to 15 copies per genome. I just want to tell you, 15 copies of K3L in the genome represents a 12% increase in genome size. This is a profound increase in genome architecture imposed by a single selective pressure from a single antiviral gene. For those of you who study recombination, what has happened is there was an initial duplication of the K3L locus, and that has now expanded up by unequal crossing over in these recombination intermediates. So what's the net result of this expansion? As you might suspect, you're making a lot of K3L protein, a lot more than you do in the wild type situation. So the vaccinia solution to facing a PKR it cannot overcome is I can't evolve a better PKR instantly, so I'm just going to make a ton of crappy PKR and try to overcome the inhibition imposed by PKR by mass action rather than by Darwinian selection. And so this appears to be a recurrent theme that is basically seen in all of the vaccinia replicates that we show. I want to point out and emphasize that the breakpoints for each of our replicates is different, which means this was not a standing variation that was acted upon. This is, in fact, a de novo duplication in every instance. We can convince ourselves that this is, in fact, what is happening because of adaptation by doing an siRNA knockdown of K3L and then infecting with vaccinia. And this just shows that in replicate C at passage 10, you're making so much K3L, but if you knock down, you can successfully bring it down significantly. And when you do that, you basically lose all of the fitness gains that you've gained over the 10 passages. So K3L is both necessary and sufficient to explain the dramatic innovation we've seen in terms of vaccinia evolution. So, of course, we don't collect box viruses from the lab which have these accordions or even from the wild. So what's going on here? Why have we basically captured this state? And one important clue comes because I want to remind you, vaccinia K3L is actually quite poor in single